Okay, can somebody confirm you can hear me?
Yes.
Thank you. All right, before we begin, are there any questions? Not hearing anything, so let me begin. Today is January 23rd, and we were supposed to be covering Lab 4 on stains. Lab 4, staining techniques. We're going to cover simple stains, differential stains, and special stains. As is normal, there is some reading to do in the textbook. This time it's a few more pages than normal, but each of these is really only about a page, maybe 2 pages. You need to read about the size, the shape, and the arrangement of bacterial cells, page 73 to 75. And I'll mention it later on in the lab. There is a difference between the shape of a bacteria and the cellular growth arrangement. You also need to read about the bacterial cell wall. That's from pages 80 to 83. You need to read about the simple stain on page 65. You need to read about differential stains. There's two of them we're going to talk about. The gram, the gram stain and the acid-fast stain. So read page 65 to 66 on the gram stain and 67 to 60, 66 to 67 on the acid-fast stain. You need to read about special stains. There's a negative stain for staining for a capsule, also called the capsule stain, and that's page 67 as well as 75 to 77. There's the endospore stain, and that's the stain endospores, and that's on page 68 or page 92 to 94. There's also the flagella stain, and that's on page 78 and page 77 to 78. There are some supplemental materials in this lab. There are video clips and a virtual lab that I hope you guys will do today. The learning objectives for this lab, you should be able to prepare a bacterial smear for staining, and we're only going to cover it because obviously you can't do it since everything we're doing is online. I should try and think of a way of getting you guys to do that in a lab question or something. Two, understand the principles and the procedures used when performing the simple stain, the differential stain, and special stains. And three, interpret the results for a simple stain, a differential stain, and a special stain. And then lastly, four, define the following terms. What's a simple stain, a differential stain, a special stain? What's a coccus? What's a bacillus? What is a spirillum? What is peptidoglycan? What is gram-positive cell? What's a gram-negative cell? What is the acid-fast stain? What is a primary stain? What is a mordant? What is a decolorizer? What is a counter stain? What is a capsule? What is an endospore? And what is flagella? Or is that several flagella? Let me think about that. What are flagella? Any question about any of that? All right. Bacteria are essentially transparent. Therefore, to see them, at least see them well, you do need to stain them before observing them under the microscope. If you were to look at unstained bacteria, they will be almost the same color as the background of the slide, meaning transparent, like the glass, which is the background of the slide, is generally transparent. Before you stain a slide, you need to make a bacterial smear. How you make a bacterial smear is you put a drop of sterile water on a clean slide, And then you use aseptic technique to transfer a little bit of bacteria into the water and then mix the drop of water and spread it, which is called the smear, all over the surface of the slide. You want to spread it not only to spread out the cells, but also to make a thin layer and then spread out the drop of water so it'll evaporate quicker. You then allow it to air dry, and then the organisms are fixed onto the slide by gentle heating. We usually call that heat fixing. You need to air dry and heat fix the cells onto the slide so they're not washed off during later procedures, such as different steps in the staining process. There's something I wanted to talk about here. Oh, if you were not to smear the water over the surface of the slide, it would eventually dry. It just would take a few minutes longer. And so if you smear the water all over the slide, it will decrease the dry time by a few minutes. Um, you only get minutes, so obviously you can't have the slide sitting for an hour because it'll air dry. In fact, probably less than an hour, probably about 10 minutes. Simple staining techniques use one dye to stain the bacteria cells. You note I crossed out the word basic because it doesn't matter if it's a basic dye or an acidic dye. It's just using one dye to stain the cells. And all you can tell, because all the cells will have the same color, all you can tell are the different shapes and the different growth arrangements, which I'll talk about both of those in just a minute. Differential staining usually uses two or more dyes to provide additional information about the cell. And the differential stain can allow you to separate different groups of cells depending on what the stain is. If it's like the gram stain, you can separate the gram positive from the gram negative cells. The gram stain is one of the most commonly used stains in microbiology, and it's one of the two differential stains we'll talk about today. Lastly, there are special stains, and they dye a particular bacterial structure, such as the cell's capsule, the cell's endospores, or the cell's flagella. Special structures within the cells. Here we are looking at both the shape of cells and the cellular growth arrangement. So here, the shape of the cell is a cocci. That is a sphere-like cell, and all of these are cocci. Here we're looking at the shape bacilli, a bacillus, also called a rod. And there we're looking at a spirilla, an S-shaped cell. Those are the three main bacteria shapes in bacteria. There are other shapes. The other shapes are not very common. There are some bacteria which are pleomorphic, and that sort of looks like a half a spirilla.
I have a curiosity question.
Uh-huh.
The upper row with streptococci, staphylococci, just the cocci and the diplococci, are
I
those all,
haven't
are,
talked about that yet. It's the
oh,
cellular growth arrangement.
forgive me. Thank you.
I'm only talking about the shape. There are cocci, there are bacilli, and there are spirilla. Those are the three main shapes that bacteria have. Bacteria can have other shapes. As I was saying, there are some that are pleomorphic, and those cells can take on essentially any different shape. There are some that are coccobacilli, and they are a cross between a cocci and a bacilli, meaning they're, I don't know, elongated cocci or very truncated bacilli with rounded edges. And then, like I said, this is like a half of a spiral. that one there. That one actually kind of looks like a spiral there. Um, but the pleomorphic can take on any shape. My point is that some of these pleomorphic are taking on the spiral type shape. Others are taking on a bacillus-like shape. Bacilli is plural, bacillus is singular. Anyways, those are the basic shapes of bacteria. You can also have cellular growth arrangements, and these are how the cells grow. Do they stick together? The first cellular growth arrangement is called the singlet, and we could have a single cocci. It's not shown here, but right here we could have a single cocci right there. You can also have diplococci, and those are two cocci stuck together. You can have tetrads of cocci, and that's actually called a tetrad. You can have 8 cells of cocci stuck together, and that's not discussed in this lab. You'll later learn it's a sarcopenia. You can have stacked phloococci, and these are cocci that clump together. And then you can have streptococci. These are cocci that form a row. So more than just 2 diplococci, you just continue the cocci in a row and it's streptococci. The bacilli can have some of these same shape arrangements. There can be diplobacilli. Actually, first there can be singlet bacilli, and that's actually what we're seeing there. And there can be diplobacilli where 2 bacilli are stuck together. There can be streptobacilli where the bacilli are stuck together on the long edge, sort of like a sausage link. Used to buy them from the store, and they're sausages hooked together one after the other. I think those are the only growth arrangements you can have for bacilli. And then the only growth arrangement you can have for spirilla is a singlet spirilla. That's the only growth arrangement spirilla have. Any question about any of this? So don't get the shape confused with the growth arrangement. I call it the cellular growth arrangement. The cellular growth arrangement is how many cells are stuck together, and you can't have a singlet. You can have a diplococci, tetrads of cocci, etc. All right, let's move on to the differential stains. The most important differential stain, and probably the most common stain in microbiology, is the Gram stain. This stain was invented in 1884 by Dr. Hans Christian Gram. This is named after Dr. Gram, which you always capitalize somebody's last name. And so the Gram stain should have a capital G in it. It is named after Dr. Gram. He used this stain as a differential stain to differentiate between the patient's cells and the bacteria cells that were causing the infection. So what he did was he stained the cells, and the patient's cells stained pink, and then the bacteria stained purple. Microbiologists started using the Gram stain, and they found that it distinguishes nearly all bacteria into 2 very large groups: the gram-positive cells, which appear purple, and the gram-negative cells, which are appearing pink in the Gram stain. The difference in the color has to do with crystal violet, the first dye used in the Gram stain, so it's called the primary dye, and whether the cell can retain the crystal violet or not. If it can retain the crystal violet, they are gram-positive cells, and if they cannot retain the crystal violet, they are gram-negative cells. Any question about any of that? All right, here we're looking at a Gram stain, a gram-negative rod, right there and there, or there, and gram-negative cocci in here. excuse me, not gram negative. That's gram positive, purple, gram positive, bacillus, and then gram positive cocci. I got the thing wrong. I got the stain wrong. I'm not doing too well today. I'm not sure why I'm so confused. But those are gram positive. And then there's gram negative bacilli right there. A gram negative bacilli, not a gram negative bacilli, gram negative spirillum right there. There are no gram negative rods that I see. Not rods, cocci. Any question about any of that? If you note I'm saying something wrong, just correct me, please. The cell wall composition differs between the gram-positive bacteria and the gram-negative bacteria. The gram-positive cell wall is structurally different. It's a very thick peptidoglycan layer in the cell wall, whereas the gram-negative cells do have peptidoglycan in the cell wall, but it's a much thinner layer, and that's the main difference between the cells. There are other differences. We'll talk about some of them today. All right, so this is a gram-positive bacteria showing you the plasma membrane, shown in green and purple here, blue, purple, and then showing you the peptidoglycan layer on top of it. This is the cell wall. In the gram-positive cells, the peptidoglycan layer is by far the main molecule in the cell wall. There are some other molecules shown like here, for example, but you can see that the peptidoglycan layer is layer upon layer. It's between 30 and hundreds of layers of peptidoglycan. And I don't know, let me blow this up a little. I don't know how many are here, but there's a layer, 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 there's a layer. And then here it's hard to distinguish the layers. But between 30 to hundreds of layers of peptidoglycan stacked one on top of the other. Very thick peptidoglycan layer in the cell wall in the gram-positive cell. The gram-negative cell, on the other hand, has a much thinner layer of peptidoglycan. Let me blow that up a little bit. Oop, too far. There's a layer, there's a layer, there's a layer, there's a layer. Probably a layer, there's a layer, there's a layer, there's a layer, there's a layer, and one or two layers on top. The peptidoglycan layer is between 3 layers of peptidoglycan and about 20 layers, much thinner than the gram-positive cell. And then the cell wall of the gram-negative cell differs because it has this lipid bilayer right here with other molecules in the lipid bilayer, but mainly the lipid bilayer. And this is called the outer membrane of the cell wall. And remember, that's the plasma membrane. You'll note that the outer membrane of the cell wall looks similar to the bilayer of the cell membrane. And in fact, most of the molecules, well, I shouldn't say most, let's say the majority of the molecules are the same. And that is the main molecule in both is the phospholipid molecule, the main molecule of the cell membrane, and the main molecule of the outer membrane. After bacteria have been stained with the crystal violet, Graham's iodine is used as a mordant. The iodine chemically combines with the crystal violet to form a larger crystal violet iodine complex. Next, the cells are treated with an alcohol acetone solution used as a decolorizer. It will remove the crystal violet iodine complex of the gram-negative cells but not out of the gram-positive cells. And that is only if the decolorizer is applied for the right amount of time. Because if the decolorizer is left on too long, it will pull the crystal violet iodine complex out of the gram-positive cells. And yet, if it's not left on long enough, it will not pull the crystal violet iodine complex out of the gram-negative cells. So the decolorizing step of the gram stain is the tricky step. It must be done correctly, and it takes a little bit of practice. The gram-negative cells decolorize, the gram-positive cells do not, and it's just because of the layer of the peptidoglycan. The timing of the decolorization is critical. After decolorizing, the gram-negative bacteria are colorless, and to see them well, you will counterstain with safranin, and that'll dye them pink. After decolorization, the gram-positive cells are still purple because they retain the crystal violet iodine complex. At least if the decolorizing step is done correctly. Now this slide is trying to show you gram variable outcome of individual cells of the same species. And if you look closely here, you can notice a difference between the purple cells and the negative cells. What do you notice different between this cell and that cell and that cell and this cell? Anyone notice a difference?
The shape is a bit different. We go from having rods to kind of, it looks more like elongated cockeye.
I understand what you're saying. It's not really elongated cockeye. What's happening is this rod has been broken right there, and that rod is broken right there, and then this rod, most of it looks like a ghost, meaning most of the cell is kind of gone right there, and so it looks ghostly. And then you have a little bit of the cell there, and even that one's a little bit ghostly. Whereas these cells, there's a very clear distinction between the inside and the outside of the cell wall. On these cells, there's not as clear of a distinction. The reason being is the gram-positive cells, the purple ones, are probably viable cells, and so their cell wall is intact. This cell wall is broken, and that's because that's a dead cell. That cell wall is dead also. And then this one, the reason why it's looking like a ghost is because the cell is decaying and it looks ghostly. Meaning in this slide preparation, we have a lot of dead cells. And as you can see, the dead cells are staining differently than the viable living cells.
So this organism would be considered gram negative then because the living cells are still purple.
The living cells are still purple, so it's called gram positive, because if it's purple, it's gram positive.
I see.
Okay. But yeah, this is probably a mycobacterium, which doesn't take up the dye as readily as other cells because it has a lot of mycolic acid in the cell wall. And every time I've had a student show me a slide like that on their species that they did the gram stain on, I've always asked them, this looks kind of weird. How old are these cells? And they've always told me that the slide used cells that were at least 2 weeks old or older. And then I tell them, okay, to get a proper gram stain, you have to only have cells that are 2 days old or younger. And the reason why you got the gram variable is because you have a lot of dead cells, and the dead cells are not staining correctly because the cell wall is decaying or broken, and then the crystal violet iodine complex can come out of the cell. Any question about that? So to get a good gram stain, you have to have your cells 2 days old at most, or you can have them younger. And if they're older than 2 days old, what you should do is streak out the cells, let them grow for a day or two, and then run the gram stain on those fresh cells. The gram stain may not work correctly if you use old cells, like this example here. And that's telling you the tips to prevent the gram variable staining of bacteria. Avoid using old cells. That's number one. Prepare a thin smear. If your bacteria smear is cells on top of cells, it's going to be difficult getting the dye down to the bottom cell. And then if it gets in the bottom cell, it'll be difficult decolorizing the crystal violet iodine complex from the bottom cells. So make sure you have a thin complex of a bacterial smear. Ideally, you want a bacterial smear that has no more than one layer of cells thick. Heat fix the bacteria on the slide only after the smear has air dried. If you heat fix the slide, and the slide cells are still wet, like water is still in there, you cause the water to boil when you heat fix the slide, and that will put holes in the cell wall. If the cell wall is damaged, the cells may not stain correctly. And then avoid overheating the cells. That will damage the cell wall. And if you do overheat the cells will look melted. Any question about that? Obviously, use fresh staining reagents. Do not over decolorize. And I can tell you, do not under decolorize as well. Any question about the gram stain? We'll talk more about the gram stain later in this lab. The acid fast stain is a stain used to stain bacteria that have a lot of a waxy substance in them called mycolic acid. These are mainly cells of two families, family Mycobacterium and family Nocardia. The family Mycobacterium has more mycolic acid in it, so it especially stains acid fast, but Nocardia does as well. If there's mycolic acid in the cell wall, the bacteria are able to resist the acid alcohol decolorization in the stain, and then the cells are referred to as acid fast bacteria positive, or AFB positive. If, on the other hand, the cells do not retain the primary stain, they will be called acid fast bacteria negative cells, and they'll have a different color. We have two acid fast stains, the Zeal-Nielsen method and the modified Kenyon method. Both of these stains use carbol fuchsin as the primary stain. Carbol fuchsin will stain the cells red. We then use a mordant to get the carbol fuchsin inside the acid fast cells. And the mordant with the Zeal-Nielsen is just applying heat. That'll bring the carbol fuchsin inside the cell wall. But with the modified Kenyon method, we have a very high concentration of phenol, which is the chemical mordant, which helps the carbol fuchsin move inside the cells. Initially, all cells take up the carbol fuchsin as the primary stain, and it's a cheery red color. We then apply the decolorizing agent, which is acid alcohol, and it will pull the carbol fuchsin out of the acid-fast negative cells. To stain these cells, because they're clear, you apply a secondary stain, which is called the counter stain, and that is usually methylene blue or brilliant green. Occasionally, a crystal violet could be used as the counter stain. I've actually never seen crystal violet used as the counter stain. At the end of the staining process, the acid-fast cells, meaning those with mycolic acid in their cell wall, will stain red, and the non-acid fast cells will stain blue or green, depending on what the counter stain was. Methylene blue will look blue, and then brilliant green, the acid-fast negative cells will stain green. So here we have the acid-fast stain applying to these cells. Let me blow this up. The greenish cells are acid-fast negative cells. They lack mycolic acid in the cell wall. The reddish or pinkish cells are acid-fast positive bacteria, and the carbonyl fusion was retained in these cells. All right, let's talk about the special stains. Remember, the special stains are a stain we use to stain a particular structure within the bacteria. There's the negative stain, which stains the capsule of bacteria. Remember, a capsule is one of the glycocalyx, a substance secreted by the bacteria, and so it's outside the cell wall. If the substance outside the cell wall, meaning the glycocalyx, can be seen with the light microscope because it has other molecules in it besides water, and it usually has a structure to it, a firm structure to it. Then we call it a capsule. The negative stain stains allows you to see the capsule because the negative stain will stain the background of the cell and the cell, but will not stain the capsule. And so you'll see a clear region around the cell, and that is the capsule. Production of capsules are associated with an organism's virulence or pathogenicity, as the capsule protects the cell from being phagocytized by a white blood cell. And that is one reason why Streptococcus pyogenes, which causes strep throat, as well as many other diseases, why a patient has a hard time throwing that disease off. And that's because the bacteria have a capsule which protects the bacteria from being phagocytized by a white blood cell. If you look at the cells in colonies on a plate, if the cells have a capsule, they usually appear like mucoid from the presence of the capsule on the cells. So that's how you look at it with your eye on the petri dish. And then capsules are not easily observed using conventional staining method because the staining method itself washes away the capsule. Therefore, you have to use a very gentle procedure to get this capsule stain, meaning the capsule stain itself is a gentle procedure. And one capsule stain is a negative stain, where you apply India ink to the slide, and then you just leave the India ink on the slide. That's shown here. India ink makes the background of the cell look dark, meaning the water, the media in the slide look dark. The capsule does not stain, and then the cell stains lightly. And that is called a negative stain or a capsule stain to show the appearance of the capsules. Here is another capsule stain. This one is using two dyes, one to stain the cell, and then a second dye to stain the background of the cell. Once again, the capsule shows up because it is clear, allowing you to now see the capsule from the cell and the background of the cell. Any question about that? Stain often uses India ink or nigrosin, although it could use any acidic dye, but usually nigrosin or India ink. And you just let the cell sit in the capsule stain, meaning this procedure has no wash step, so you're not going to wash off the capsule. The same with this procedure. This procedure, if I remember correctly, you apply the dye, let it air dry, and then you apply the second dye to stain the background. And you then can either let it air dry, or more often people just view it under the microscope with the wet second dye on there. And either way, you can see the capsules. Capsules, as I stated in the lecture, you will not be tested on it really in the lab other than perhaps this lab. And you do need to know about it because your unknown project may have you choose to do the capsule stain to look and see if the cells have capsules. Any question about any of that? All right, if not, then the last stain to discuss, at least I think it's the last, is the endospore stain. Endospores are a metabolically inactive structure that is hardy in that it allows the cell to survive even in unfavorable environmental conditions. So when the environment becomes favorable again, the endospore will then germinate and the cell will change from an endospore to parent bacilli. And the parent bacilli will have the normal metabolically active state, also called the vegetative state, and the parent cell or the vegetative cell can reproduce. The endospore is metabolically inactive, and what it did is it shuts down metabolism to almost nothing. It isn't quite nothing, but it's very, very low metabolism. And the endospore allows the cell to survive the harsh environmental condition, which will be harsh enough to kill all of the parent vegetative cells. Most endospores are made from two genera, the genus Bacillus and the genus Clostridium. So these two families. Most endospores are in these two families. This table is showing you some of the bacilli that form endospores that are clinically significant in humans. There's two bacillus, Bacillus anthracis, which causes anthrax, and Bacillus cereus, which causes the disease of food poisoning. And then there's Clostridium botulinum. This species has an endospore, and the species can cause food poisoning, more often called botulism. Clostridium tetani can cause tetanus. Clostridium perfringens can cause gas gangrene and food poisoning. And then Clostridium difficile can cause antibiotic-associated colitis, which is usually diarrhea the patient gets after taking antibiotics. The antibiotic can kill off the other bacteria, but it does not kill off the endospore of Clostridium difficile. And then when the environment changes and becomes favorable for the bacteria to grow, the bacteria come out of the endospore and then grow, and that would cause the patient to become sick again with diarrhea. Antibiotic-associated colitis. Any question about any of that? All right, to determine the endospore, we use the Schaefer-Fulton endospore stain. It uses malachite green as the primary stain. Malachite green normally dyes both the vegetative cells, like the parent cells, and the endospore. So initially, they're both green. Heat is then applied as a mordant to help the malachite green get inside the endospore. The cells are then decolorized with water. Water will remove the malachite green from the vegetative cells, including the parent cells, but it will not remove the malachite green from the endospores. Safranin is then applied as a counterstain to stain the clear cells, which became clear after you washed out the malachite green. Come on, mouse. Having a hard time finding it. There it is. Here is a stain, staining for endospores, and it's the Schaefer-Fulton stain. And let me blow this up. The vegetative cells are pink, like that one and that one. These are the metabolically active and reproducing cells. That's an endospore outside the cell. It's come out outside of the cell, and you can see it's green. And there is an endospore inside a parent cell. The parent cell makes the endospore, and then when the parent cell dies, the endospore is given off, and that's how those form. Any question about any of that? All right. So understand the term flagella staining. Let me state that if the cell can move or swim, then that bacteria cell likely has a flagella, because the flagella is the only structure that bacteria have that allow the cell to move or to swim. Flagella are composed of the flagellin. It's a class of protein. You can also classify the flagella on the distribution of flagella on the bacteria surface. Oh, come on, mouse. I'm not seeing the mouse for some reason today, unless I get it directly on the thing. So bacteria, if they have one flagella, it's called monotrichous. And bacteria could have no flagella, in which case it's none. And if there's multiple flagella on one end of the bacteria, it's called lophotrichous. If there's multiple flagella on both ends of the bacteria, then it's called amphitrichous. And if you have flagella all over the bacteria, coming out almost in random position, then it's called peritrichous. You're not going to need to learn the distribution of flagella for any quiz. You only need to learn it for one lab question in this lab. Any question about any of that? So I'm not going to quiz you on the flagella arrangement. Flagella are too thin to be seen with standard staining techniques, so special stains are required to view the flagella under a microscope? Flagella stain. Let me blow that up as much as I can. What do you notice about some of these cells compared to other cells? Like what's different between this cell and that cell, or this cell and that cell?
The number of flagella surrounding the cell or just of one end of the cell.
Yeah, the arrangement and number of flagella are the differences. So this is all cells of one species. And the reason why this cell doesn't have any is because this cell probably just divided. And once a cell divides, initially the cell will not have any flagella because the cell has to make the flagella. And this cell was recently born, so to speak. We recently came through cellular division. So it has made no flagella yet. You have to give it time for that. And so if we were to look at this cell sometime later in life, it would put out a flagella on one end first, and it would be monotrichous. And then it would add more flagella to one end, and it would be lophotrichous. And then it would add more flagella, and they will come at the other end, and it's called amphitrichous. And then, if given enough time, the organism might get a trying to remember the name of that word, the drawing a blank now. Um, well, the cells can have different flagella depending on where the cell is in its development. Initially, the cell will have no flagella, and then it will put out one flagella on one end, and that will be monotrichous. And then it'll put multiple flagella on one end, it'll be lophotrichous. And later we look at it, it'll put out flagella on both ends, and it'll be amphitrichous. And then later when we look at it, it can put out flagella everywhere, and it'll be called peritrichous. So how do you name these? If in this case you see multiple flagella arrangements, which one do you choose? Anyone want to guess?
Do we go ahead?
I'll take that as a no, but actually you were going to say something. If you want to say it, go ahead.
Um, Predominantly, would we go with, the Lophotrichus just because it's the one we see most in the picture?
That is a logical conclusion, but no, you don't go with the one that has the most. For one thing, it's really difficult to determine in the picture which one has the most. Do you think Lophotrichus is the most? Maybe, I don't know. I have to look. No, you go with the highest order
Okay.
because given enough time, the cell will become peritrichous. So we say that species is peritrichous in its flagella arrangement. But some cells, the highest they can become is amphitrichous. So you'd say amphitrichous is that species. And then there's another species, the highest they can get is lophotrichous. So there you say that species has a flagellar arrangement which is lophotrichous. And then there are some species that only has one flagella, and so that species will be monotrichous. And the only diversity you'd see on this one is there'd be cells with no flagella and then cells with one flagella. And of course, there's also species which has no flagella, and that's actually the majority of bacteria species. They do not have a flagella, and they'll never have a flagella, assuming they don't evolve one. So when you see multiple, and usually you will see multiple because the cells will be in different developmental stages. You call the stage by its highest order. All right, flagella are too thin to be seen with standard staining techniques, so you need special stains to view the structure under the light microscope. And with the flagella stain, most of them, what they do is they just, let me get the picture again, they just coat the flagella as well as the entire stain with multiple coatings, and then you can start to see the flagella. And with the just one coating, you won't be able to see the flagella because there's not enough there to view. All right, know the terms of this lab. You can learn, go through the references if you want. I wouldn't bother doing that. Let me go to the worksheet now. Oop, didn't get it. All right, you're not going to be doing anything in this lab because everything we do will be done online. However, there are some videos in a virtual lab that you need to complete to understand this lab. And some of the questions, at least one of the question, comes from the virtual lab. Anyways, we have some video clips for you to watch on preparing a bacterial smear, and then later a video on heat fixing, air drying and heat fixing. And then these steps go through the procedure for making a smear. Let me iterize that a smear is something you do before applying any stain. Most of the time, when you're going to stain a bacteria slide containing microbes, you will make a bacterial smear or a microbial smear first, and then you'll take that smear and stain it. So the staining is not anything to do with the smear. They're independent events. And it is true to do the stain, you essentially need a smear first. But to do the smear, you never have to do the stain. I don't know what would be the purpose of that, but you don't have to do the stain. So read through the steps of making a stain. We have 2 videos for you to watch: preparing a bacterial smear and then heat fixing. Oops, too far. Then scroll down to the question of procedure 1, 3. So procedure 1, question number 3. What are 4 common errors when preparing a bacterial smear for staining? It has to be an error in the smear. It cannot be an error in the staining. Any question about that? All right, for procedure 2, we're going to look at a simple stain. So here's a video clip of a simple stain. And just review the steps given here for undertaking a simple stain. You first get a bacterial smear, and then you add dye to the slide. And after about 1 minute, you wash off the dye with gentle, clear water, cold water. I shake off the water, blot the slide dry. I've showed you that in the lab, but let me go ahead, not the lab, the lecture. You get a piece of bibulous paper, and I would say rip it out of the book. Fold it and fold it over your slide. I don't know if you can see this. I'm trying to show you. So this is my slide. Get the bibulous paper and fold it over the slide and then press down gently. And that will remove the majority of the water. It will remove enough water that you can even apply oil and then look at the slide under the microscope. Oh, some important parts with that is do not take the bibulous paper and rub it against the slide because that'll rub off cells. Likely, also don't take the slide and then rub it against the bibulous paper because that will remove cells from the slide. And then answer the questions to procedure 2. Procedure 2A, what is the cellular morphology? That is the shape, not the cellular growth pattern or growth arrangement. So E. coli is shown here and Staphylococcus aureus here. Let me let me inform you that this is a clumping preparation of cells, and you have to note where the cells are and where they are not. Come on, get out of here. So here, that's one cell there, and that's one cell there. This looks like it's actually 3 cells. One cell there, one cell there, and then one cell there. This one is actually 3 cells, one cell there, one cell there, and then one cell there. That's 2 cells, one cell there, one cell there. So unfortunately, in this slide preparation, the cells are sticking together or clumping together, and you have to remove the cells that are clumped or ignore them when you're trying to determine the shape of E. coli. And then you can get the shape of the Staphylococcus aureus just by looking at the cells. And then fill in your answer for the shape for E. coli and the shape for Staphylococcus aureus here. And that's procedure 2A, 1 and 2. And then procedure 2B, answer the question, what is the total magnification that is used in this lab? Any question about any of that? For procedure 3, we're going to be looking at the gram stain. We have a video clip of performing the gram stain, and we have a virtual lab on the gram stain. When you open this virtual lab, it may take a little while to download depending on how fast your internet connection is. I've already downloaded this in 2:30, and so that didn't take any long time. Well, this will be a virtual lab on the gram stain. I'm going to shut it off and then just move it forward. You need to go through the procedure here. I'm only going to go through the gram stain. So in this virtual lab, wherever you have the arrow, you need to pull this down to here and follow the arrow. And that's how you light the Bunsen burner in the virtual lab. Once again, follow the arrow. So you take this and go down. And that's the first time. Oh, I thought that one took more than one, but apparently it only took one. Just trying to speed up. All right, performing the gram stain. We put the bacteria on this slide here. I'm not going to go through the steps. You should really do it with the loop and get it everywhere. Heat fix the cells after they are dried. Here we finally have the gram stain. Let me turn on the sound.
Specific order and rinse the slide off under slow running water in between each chemical. The first is crystal violet. This is our purple stain, which will indicate gram-positive bacteria. Using the dropper, squeeze out enough drops to cover your sample. Let that sit for 60 seconds. Now rinse it under slow running water. Now use the bottle of Gram's iodine. Gram's iodine will fix the dye to the cell walls of the bacteria and make it permanent.
If it's gram-positive.
Squeeze several drops onto the sample, enough to cover it completely. Let that sit for 60 seconds. Now, rinse it under slow-running water.
All right, that's how you go through the gram.
Now we are
procedure.
going to use the alcohol solution.
So you guys
This
can,
will wash off the excess dye, leaving only the dye.
so you guys can finish this virtual lab. Any questions about the virtual lab? So go through the virtual lab because that's the only way you can actually get practice on performing the gram stain in this class. All right, this is the steps for the gram stain. Review that. Initially, you put crystal violet on the cells, and all the cells will stain bluish purple from the crystal violet. So both the gram negative cells and the gram positive stain bluish purple from the crystal violet. Then the next step is applying the iodine. Iodine is a mordant. It complexes with the crystal violet. So the intensity of the cells, or the intensity of the dye, should increase, as well as the fact that the cells will change in color slightly. Initially with crystal violet, the cells are a bluish purple. After the iodine, the cells are more of a purple purple. Very little change in color. You'll note there's no change in the intensity of the stain or a change in the color, but trust me, there's a slightly more intense stain here. It's barely noticeable, but usually you can notice it, and you don't always, but usually you do. And then there's no change in the color. Like I said, there is a slight change in the color. And then you apply the decolorizing agent that will pull the crystal violet iodine complex out of the gram negative cell, so they will appear clear. The gram positive cells will retain the crystal violet iodine complex, and they will retain their purple color. You then stain with safranin. It's the counter stain, and it will get the gram negative cells staining pink and the gram positive cells staining purple. And then fill in the answers to this table. Give me the gram reaction, whether gram positive or gram negative, for E. coli and its cellular morphology. Is it a coccus or a bacillus? And then do the same for Staphylococcus aureus. Give me the gram reaction and then the shape of the cell. Any question about any of that? Then do procedure 3A. Tell me the color of Staphylococcus aureus after the primary stain is added. And the color should be pink, purple, or colorless. And then give me procedure 3B. E. coli, what's its color after the primary stain is added? Write it here. C. Staphylococcus aureus, what color is it after the alcohol acetone step is performed? Wrote it there. And then D. E. coli, after the alcohol acetone step is performed, what is the color of these cells? And write it here. And then Staphylococcus aureus, what color is it after the counter stain is added? Write it right there. And then E. coli, after the counter stain is added, what color is it? Put your answer right there. Any question about any of that? All right, then we have the acid-fast bacteria stain. This is the second differential stain that we're going to talk about. So we've already talked about the gram stain. Let's talk a little bit more about the acid-fast stain. First, view the clip on the acid-fast bacteria, and then review the steps for performing the acid-fast stain. We're going to be looking at the modified Kenyan acid-fast bacteria stain, and that's what the video covers, as well as this lab. So first you add carbonyl fusion. That causes the acid-fast positive cells to stain pinkish, and the acid-fast negative cells also stain pinkish. You then decolorize the cells with an acid alcohol wash that will remove the carbonyl fusion from the acid-fast negative cells. So at this point, those cells will be clear. It will not remove the carbonyl fusion from the acid-fast positive cells, so they will stain pinkish. You then apply the counter stain, which in this case is brilliant green. The cells that have that are acid fast, meaning they have mycolic acid in their cell wall, will stain reddish or pinkish. And then the cells that lack the mycolic acid in their cell wall will be acid fast negative, and they will stain greenish or green. Any question about the acid fast stain? All right. If not, we can come down here to this picture and answer the questions denoted by the arrows. If the cell is acid fast positive, acid fast bacteria positive, or acid fast bacteria negative. You should have an answer at each arrow, and you can write it like right here and right there, right there and right there, or you can type it right above the picture or below the picture and say in the picture on the leftmost arrow is tell me if it's acid fast positive or negative, and then the righter picture or the higher picture, excuse me, not picture, the higher error or the rightmost error in the first picture, tell me if it's acid fast, positive or negative, write it there. And then talk about the modified Kenyan method or the second picture. Tell me if the arrow on the left is acid fast positive or acid fast negative, and then tell me the same for the arrow on the left. Any question about procedure 4? All right, in procedure 5, you're going to be looking at a video clip of an endospore stain and a negative stain. And then we have 3 pictures here of 3 different bacterial smears which have been stained and then observed at 1000x total magnification. Picture A refers to question A, picture B to question B, picture C to question C. So the first question in procedure 5A, so A1, is this is the Schaefer-Fulton stain, which uses what as the primary stain? So name the stain here. And ultimately, that stain dyes which structure of the cell? And the structure are produced by some bacteria to aid in survival. So that would be procedure 5A2. Procedure 5A3 then asks, what are some examples of pathogenic endospore producing bacteria? And in the lab module, you're given some examples. Excuse me here. My fan just turned on, so I'm getting a little stuffed here. You need to give an example from the lab module, because if you give me an example from AI, meaning you simply say, what are some examples of the Schaefer, Fulton, pathogenic endospore producing bacteria, the AI will give you an answer, but I don't want that answer. I want an answer coming directly out of the lab module. And I think the answers will be the same, but if they differ and you give me an AI answer, I'm going to mark it wrong. So make sure your answer comes out of the lab module. In procedure 5B, referring to picture B, this is a name the stain that coats long whip-like appendages which aid the cell in motility. So this is the stain here. So name the stain. And then in 5B2, state the flagella arrangement type that you see in the picture. And then in 5BC, what are some examples of clinically significant bacteria that produce flagella? Once again, if you give me an AI answer and it's different from the lab module, I will mark it wrong. So make sure you get your answer from the lab module. So for procedure 5C, question C1, this is what's the stain, name the name of the stain that uses India ink or nigrosin and can be used to detect the presence of a cell structure, name the cell structure, which is located exterior to the cell wall and prevents phagocytosis. So C1, name the stain. C2, name the structure discussed in C2. And then for procedure C3, what are some pathogenic microorganisms that produce capsules? Once again, you must give an example that's been given in the lab module. Obviously, you could get that from AI as well, but if it differs, I'm going to mark it wrong. Meaning I really don't want you to answer this using AI. I want you to answer it by looking it up in the lab module. All right. Case studies. Case study 1. This is a 45-year-old female experiencing respiratory issues. She's having trouble breathing. Based on her clinical presentation, the doctor orders a chest x-ray, shown here, and then they collect sputum for an AFB culture. In addition to the culture, an AFB smear is made and stained. Based on the findings below, what is the most probable diagnosis? And you must name the species which is causing the infection. In the x-rays, normally the lungs appear dark, like right here. This right there, this whole region, and that region there, and then this region here, something's going on. The lungs in that region there, the lungs do not look correct. And then that's the smear, and then there's the stain of the smear. They did the Zeal-Nielsen stain. So tell me what the species is depending on this information. The species causing the patient problems. So you must name the species. You can't tell me the disease. Case study 2, we have a 64-year-old diabetic patient who's admitted to the hospital with soft tissue necrosis. There's the soft tissue necrosis there, and it looks really bad. A routine culture and gram stain is ordered. The bacteria grew aerobically. Based on the findings below, what is the most probable diagnosis? You must name the species which is causing the infection. You can't simply say diabetic soft tissue necrosis. I had at least one student do that in the past, and that's an incorrect answer. You must give me the bacteria which is causing this, And obviously there's the bacteria there in the gram stain. And there's the bacteria here in the Schaefer-Fulton stain. And then the bacteria are growing aerobically. So name the species which is causing this. Any question about the case studies? If not, go ahead and work on the lab. I will be here until at least 8:10 to help you with any questions in the lab. Or I will be here until the last student logs out. So if you have a question, make sure you ask it before the last student logs out. Any questions?
Yes. Using the lab module, does that also include the page numbers up in the pre-lab textbook readings?
You do not need to answer with the page number.
Okay.
Okay. I'm not sure, but you can use the lab module, anything in the lab module, to answer these questions. All right. Any other questions? Okay. Go ahead and get started on the lab. I will be here to answer questions until the last student leaves. I may step out in a little bit to go to the restroom, but I'll return in about a minute or two.